Good morning, everybody. So welcome to this Monday seminar. It's our great pleasure today to have Emma Robinson with us. So Emma is a lecturer at the King's College London, and she focuses on the development of computational methods for brain imaging and neuroscience, and now also increasingly AI methods. So of course, you know well the very important contribution of Emma on the multimodal surface matching technique, which has been particularly important in the success of the HCP data analysis that has spanned the last decade, I would say. And so it's very a great achievement that has been done with your contribution on the precision matching of cortical surfaces. And so now currently you are developing some geometric deep learning tools for modeling functions on the cortical and cardiac surfaces too. And also optimizing image processing pipelines, open data collections, etc. So such as HCP and the UK Biobank, and building a learning-based model for neurological process analysis, so neurological digital twins. So it's really nice to have you here. We're going to listen to your talk on precision cortical imaging. Thank you also to Jessica for organizing the visit. And so we have the presentation and then some questions from the audience.
Okay, thank you very much. I'm really pleased to be here. Yes, thank you for having me. So today I'm going to talk about work that I do in my lab to try and improve the precision with which we can compare cortical imaging data across populations in order to better understand neurodevelopmental and cognitive phenotypes. We are taking a two-pronged attack to this, so we're trying to improve both classical methods based on image registration and increasingly learning-based methods for reasons that will become apparent as I go through my talk. So obviously, as we all know, the cortex is So as we all know, the cortex is the thin layers of tissue at the outer surface of the brain, which are much expanded relative to our non-human primate cousins, which implicates in a wide range of higher order cognitive processing, which is why we're interested in it. And the process of cortical development is complex and multifaceted, but if we break it down into very simplified terms, it involves the birth and the proliferation of cells at the center of the brain within the ventricular zones. They then migrate out radially towards the cortex along the scaffold of glial cells to form the 6 layers of the cortical plate in an inside-out sequence. And then when the cells reach the cortex, they start to form intracortical connections, which results in a much greater rate of expansion of the superficial cortical layers relative to the inner layers and the white matter tissue below, which creates biomechanical tensions, which result in folding. And this doesn't occur in a uniform way, but rather follows a well-characterized schedule, starting with the formation of the central sulcus and sylvian fissure before 24 weeks, before then progressing to the rest of the primary folds, secondary folding patterns such as the superior temporal sulcus, before entering a period of tertiary folding just before birth. And this then continues, but much of your cortical shape is fixed by the time that you're born. We are able to investigate the trajectory of cortical surfaces, or we've done this in the past using multimodal surface matching, or MSM. So MSM is a spherical registration algorithm in the sense that the easiest way to align different brains is to project the surfaces to a sphere and then match imaging properties on that sphere that in this case reflect the sulcal depth. But because you have vertex correspondence between the spheres, that's not moving, the spheres and the Because you have a vertex correspondence between the cortical anatomy and the spheres, it means that you're then able to learn an implicit walk between the cortical anatomies and regularize that biomechanically in order to learn a biomechanically constrained longitudinal mapping. And this was used by a collaborator of mine, Cara Garcia, in 2018, with a very unique cohort that they had at WashU, which were 24 babies born very preterm and then scanned at regular intervals between 28 and 38 weeks of birth. So longitudinal biomechanically constrained registration was run within subject. They were then all mapped to a population average space so that we could compare those growth trajectories statistically. And this showed that while cortical growth was initially fastest around the somatomotor areas, it was this is later overtaken by growth of higher order cognitive regions in the temporal lobe and frontal areas. And it's possible to also compare these patterns of growth cortical expansion to gene expression atlases. So this was a recent paper by Gareth Ball, in which he created his new brain atlas, where he took the fetal histology from the Allen Brain Atlas, repaired any of the data that was damaged during collection using a pix-to-pix generative model, used slice-to-volume reconstruction to put these histological slices into a 3D volume, fit a cortical surface to it, and then compared it against cortical expansion maps that were obtained from the DHCP fetal surface templates generated by Slava Karolis. And at the same time map gene expression data onto that surface, so that he was then able to identify 433 what he calls zone region time point genes that are implicated in aerial scaring in gestation. But what we know is that errors at any stage of this process of cortical development results in well-characterized malformations of cortical development. So we have microcephaly, which is very extremely small brains, megalocephaly, extremely large brains, heterotopia, where some of the neurons fail to fully migrate to the cortex, so you get a band of grey matter within the white matter, lissencephaly, overly smooth brains, polymicrogyria, so an overabundance of shallow folds, and focal cortical dysplasias, where these are very subtle, these appear very subtly on structural MRI. There's different types with different causes during development, but all are associated with drug-resistant childhood epilepsy. So it's really important to be able to find these, but they can be very difficult to see. But perhaps, with the exception perhaps of polymicrogyria and focal cortical dysplasias, many of these MCDs are already visible on an ultrasound. So a stronger argument for why you might want to use MRI would be for those cases where the neurodevelopment of that fetus or neonate is more difficult to predict. So a strong example of this historically has been preterm birth, where people have been doing studies on preterm birth for extremely long time now, because you're able to image these babies when they get out of the womb. And we know that all of these individuals are associated with long-term neurodevelopmental impairment, but it's much more difficult to predict the individual outcome because their neurodevelopmental trajectories are so heterogeneous. Nevertheless, in this study by my PhD student Kylie, she looked at the morphological signature of preterm birth across the lifespan using 3 cohorts. So she has the DHCP cohort, which has 541 babies born healthy at full term and 81 very preterm neonates scanned at term. There's also the BIP, which is a follow-up of an older study of very preterm babies that were then later scanned in childhood and compared against childhood controls, and UCLH data, which is preterm individuals scanned during adulthood, and found a consistent signature of cortical impairment consistent signature of differences in cortical morphology, with extensive differences in cortical surface area, reductions in sulcal depth and thickness that are consistent across the lifespan. We've started to see whether we can compare these against the fetal data that we've acquired from the DHCP. And what you find is that there's evidence of differences appearing in those areas of the brain that are growing or developing in those later stages of development. So here we have fetal data and neonatal data that were acquired at very different starting time points. So it's a bit more of a difficult, more statistically low-powered study than the one originally done by Cara, but nonetheless, we're then able to compare healthy brain growth in utero against ex utero development, seeing that there's significant differences in these areas of the brain that are growing latest around the parietal temporal junction and the frontal lobe, with the preterm showing elongated brains because they're lying with their brains on. They're lying with their heads on the side of they're lying on the side of their heads in the incubator. But there's some evidence of these preterm babies starting to catch up within the insula, for example. And there was a follow-up study by Cara Garcia that was published recently, who followed up that very preterm cohort into childhood and showed again that many of those areas of the brain that are less developed at birth are growing faster all the way into childhood, but nonetheless, as we've seen from Kylie's study, they never fully catch up. And what's interesting is that those areas of the brain that don't fully catch up, if you take their surface area and sulcal depth and run a canonical correlation analysis with outcome measures and clinical factors, you find that the differences are correlated with the clinical risk factors, for example, how long a baby was kept in ICU, as well as their cognitive outcomes at 20 months. So this was done from the E prime cohort. So these babies were predominantly very preterm. And you see that it's correlated, but the outcome data is noisy. So it's very difficult to predict individual outcomes. Try not to break them. So this suggests that these areas that are being picked up as consistently different could potentially represent a potential biomarker for therapeutic intervention. And interestingly, this pattern that those areas of the brain that are developing latest are most impaired is also seen in babies that are born with congenital heart disease. So while this is extremely promising at the population level, suggesting that there are consistent differences between these cohorts and that this is correlated with outcomes, nonetheless, it's still extremely difficult to predict the individual outcomes of these babies. And part of the reason for that is the scale of heterogeneity of cortical neurodevelopment. And we have a similar problem in aging, and then it becomes very difficult to determine what is normal for a specific for any given individual, and that's the reason why it's so difficult, I think, with imaging data to reliably predict outcomes. And so for this reason, we've been looking at characterizing in fine detail the scale of variability of cortical folding within the healthy adult HCP cohort and the DHCP cohort. So this was work by Yuan Guo. And she used learning-based cortical surface registration because this was running so fast. She was able to register all pairs of hemispheres of the young adult HCP and DHCP together, and then cluster them into groups from which she could generate 30 group templates, which characterize in sharp detail all of the modes of cortical shape variability for each of the frontal, parietal, occipital, and temporal lobes. She did the clustering separately for each because she visually observed quite a lot of variability and lack of co-occurrence of cortical folding patterns across lobes. So just because some individuals share a pattern of cortical folding in the frontal lobe does not mean that they necessarily share cortical folding patterns in the parietal, occipital, or temporal lobes. And once she'd got those templates, she then co-registered them to get everything into a population average space. And why we thought this was advantageous is because it then gives you a straightforward way of really visually contrasting these different forms of these different motifs of cortical folding within the same space, to characterize them in fine detail, study statistically their patterns of co-occurrence across lobes, look at across individual variability and how this relates to heritability, because both of these cohorts are made up of quite a lot of twins. And more importantly, once everything is aligned through image registration, then we can start to compare this morphological variability against other imaging properties, for example, cortical function. So the result was that is 30 sharp templates that show significant variability in terms of the orientation, splitting and branching of folding patterns. She statistically analyzed co-occurrence and didn't find strong evidence to back up her assumptions in the way that she built her model. Also looking at genetic heritability, although genetically identical individuals have more similar patterns of cortical folding, they are not exactly the same. And one of the big advantages of doing it this way is then we can start to look at more detail about what properties of cortical morphology are informing function and which are not. So she looked in detail at the asymmetry of these folding motifs. So because the hemispheres had, the right hemispheres were flipped to left and they were all co-registered, it was possible to look for which templates were more leftward dominant. And she found that there was significantly more asymmetry of the folding templates of the temporal lobe, with temporal lobe variants showing significantly greater complexity, which aligns with known laterality of language in this area. They also have greater surface area. But when we looked in more detail about the extent to which cortical function was aligned with these templates by trying to see, our original assumption was that if we did a much better job of aligning cortical folding in areas that were really variable, that surely we would do a better job of aligning function, and this was absolutely not the case. So if you look within cluster, task activations become slightly worse overlapping when you do a better job of aligning folding patterns, as do the cortical areal parcellations. And interestingly, so the morphological features, the morphological results were replicated in the DHCP. So the DHCP, so at birth, these babies show a similar degree of variability, though slightly reduced. So in general, she was able to match each neonatal template to approximately 2 adult templates. But the asymmetry was also present, but less statistically significant, which makes sense because the brains are still developing. But overall, our feelings through these analyses was somewhat that it appears that away from primary folding patterns, it's possible that secondary and tertiary folding perhaps is driven somewhat by random instabilities during the cortical folding process, driven perhaps slightly random differences in the rates of cell proliferation or migration. That's because as you proceed further and further into development, this process is more unstable. It is generating folds in some individuals that are differences in folding patterns between genetically identical individuals. And that in some senses, perhaps these variabilities in folding are just something of a distraction. And that when it comes to functional organization, this is of course, the variability continues to be much more impacted by environment and plasticity after birth. But in some senses, this study raised more questions than it resolved. And the problem with all of this, of course, is that in the long term, or the way that we classically analyze all our imaging data is that we try and map it into a single population average space in which we assume that all of common structures, whether it be folds or cortical areas, are perfectly overlapping. and that we know that all of our cortical properties vary at sulci, whether you're in a sulcus or a gyrus, both to do with both for biological reasons, but also for image processing regions. And this then creates noise in downstream analysis. So a few years ago, she looked at this in the first instance by building a sort of high resolution normative modeling of cortical folding development in the neonates. And what she observed was what looks like outliers of your normative model just happens to relate to areas where some individuals happen to have a rare gyrus, where others would have a sulcus. And this is then a problem because this small amount of image processing noise stands to obscure subtle signatures of cognition and disease potentially. But this was all motivated by the fact that our long-term goal was to try and find these subtle focal cortical dysplasias. And when you're talking about subtle differences in cortical focal depth or blurring of the white matter boundary, these sorts of elements of noise are quite a problem. So for this reason, having done our absolute best to improve overlap of cortical features through classical image processing pipelines, I have in parallel been investigating deep learning methods, and that's because the advantage of deep learning is that you can train these frameworks to be transformation invariant. And part of the reason for that is that 2D and 3D convolutional neural networks are translational equivariant, and that's because which basically means as you translate your window across an image, the output of the convolutional operation translates with it, and that means that convolutional neural network can be trained to be translation invariant, meaning they can identify an object irrespective of where it is in the scene. So you can identify a lesion irrespective of where it is in the brain, in principle. The problem is that you still need lots of data to do this in order to learn invariance to all the other types of transformation that you might observe in the data, and that in their original form, deep learning frameworks are black boxes, which can be difficult to interpret, so you don't know why they make the decisions that they do. So there are two ways of dealing with these. The first is that you can improve your ability to train in the low data regimes that we have within neuroimaging and medical imaging data by using augmentation. So you can use image registration to remove as much variability as you can, and then you can simulate the different types of variability that you expect to see in your data in order to make your model robust to those types of variants. And I just have a figure up here from MIT's SynthSeg paper, because this is probably the one that does it the best. So they have developed brain segmentation methods that are robust to any type of acquisition or contrast by using a generative model which synthesizes from segmentations all the types of variability that they expect to see in the data. And then they have a model that performs robustly on different types of contrast. So this is an engineering solution to that problem that works to an extent. The problem then becomes, how do you build a learning model that is human interpretable? And this is perhaps a more difficult problem. So the first thing that people used to do was to build methods that had post hoc explainability. So they would generate saliency masks. So these basically take the signal that you're getting out of the network, so the activation of the output neuron, which determines some classifications. In this case, it's trying to classify Alzheimer's disease from mild cognitive impairment, and then you project it back through the network in order to predict what features of the image were most important for driving that prediction. And the problem is that because convolutional neural networks downsample the data through the network, those original predictions are very low resolution, and they're also not fully discriminative. So for a classification, all you need to do is pick up on the most salient features that are discriminating between classes. So in the case of Alzheimer's disease, all they have to do is detect that the ventricles are expanding, and not even the full ventricles. They're just choosing to look at this particular part, and that's sufficient to distinguish Alzheimer's disease from MCI or mild cognitive impairment. But in an ideal world, from a clinical explainability position, what you want to know is where all signs of disease. You want to be able to pick up on all signs of disease within that image, or all features within that image that are predictive of a particular behavioral cognitive phenotype. So this is not great. Better are generative models or conditional generative models that will give you an explanation for a specific class, or use of self-attention models from transformers, or even better, models that are built to offer a causal explanation of which particular properties of an image are predictive of that are predictive of a particular disease type, for example, or to build learning-driven but physics-constrained models. So I'll talk about all of these as quickly as I can. So the first method that we looked at to try and improve or the ability to understand and pick up on all features of disease was ICAM. So this is a generative model that performs image-to-image translation. So what that means is it will take an image that has Alzheimer's and learn how to translate this back to a prior state of mild cognitive impairment. In the process of having to do that, it has to pick up on all features in the image that are predictive of that disease. But we go further than just doing that to embed the model with some understanding of the biophysical constraints, which is that we know that a lot of the variability is confound variability that we need to disentangle. So we learn two latent spaces of a generative model, one large latent space, which is modeling the cortical shape variability, and another smaller attribute space, which is learning the phenotype of interest. And this is a variational autoencoder, so that means that we're able to sample from this space, and we can then take the content latent of each image, swap out its attribute latent to one of a different class, regenerate the image, and it gives us a difference map that picks up on all signs of disease within that individual image. So here you can see that ICAM is doing better than a conventional image-to-image translation model, which isn't embedded with this specific disentanglement constraint. So this is just learning to trying to learn to translate the image without understanding that there is nuisance variability. And in doing this, the ICAM is able to pick up much better on all signs of cortical variability or cortical atrophy. And they're both doing a much better job than these post-hoc saliency methods that are just generating a false explanation. We can do the same for surface convolutional neural networks. So here we're learning a difference map which can smoothly change the age of a baby. So essentially we're learning a deep normative model of development. So here we're making a slight assumption here that preterm babies' first scans are healthy. We're then training the model to smoothly transition the age of each individual. It does so well enough to completely fool an independently trained regression network. And when you look at the deviation between the ground truth follow-up scans of preterms and their simulated healthy scans, you do a bit better about being able to predict the cognitive outcomes of these babies at 18 months. I should say that doing deep learning on the surface is somewhat non-trivial, and that's because much of the power that you get from convolutional neural networks comes from these properties of translation equivariance, which means that you don't have to learn this variability. And this is exactly what we're trying to get at, right? We're trying to learn the residual variability that we can't model with image registration. And this is very easy to compute. This is very easy to compute because you're just taking out patches of the images which reflect a convolutional filter and performing a matrix multiplication with the image patches at this point. And then it becomes very computationally efficient to learn these parameter efficient networks that can learn to recognize these objects wherever they are in the scene. And any other invariances we need to learn through augmented training, through learning even more image filters, which then puts at risk of overfitting and poor generalization. So we need to embed this idea of rotational invariance into our surface models, where rotation would be the equivalent of translation for a sphere, but this is more difficult to get because in manifolds we have no global coordinate system, which means that any path that we were to rotate a filter across the screen in order to spatially learn this convolutional operation is path dependent, meaning that the orientation of a filter, if it starts here, would flip left, right if we moved it along the equator, but would flip upside down if we moved it on the over the top. So this means that it's not rotationally equivariant, it's gauge dependent, which is a bit of a problem. And learning spherical surface deep learning frameworks that are rotationally equivariant is for that reason non-trivial. You can do it in the spectral domain. So here you have the convolutional theorem, which says that the Fourier transform of a filter and an image, the Fourier transform of the convolution of a filter and over an image is equal to the product of their Fourier transforms. And this is easier to generalize to the surface domain because we have a we can generalize the Fourier transform to the sphere using, for example, spherical harmonics, or to a general manifold using Laplacian bases. But doing this, we learn non-local filters that, and the operation itself is computationally really heavy because you have to do matrix multiplication of large, you have to do eigendecomposition of large matrices. So what people do in practice is that they make approximations again, and they end up a conventional graph convolutional neural network is using very simplified, rotationally invariant filters that have no expressivity. So in short, basically trying to develop computationally efficient convolutional neural networks is really difficult, but what you can instead do is use vision transformers. So vision transformers were developed in 2020. They take the idea of the transformer model, which was used to originally for language. So in language, the idea is that you learn a model that learns how different words in a sentence or in a paragraph attend to each other in order to convey meaning. So you have very long range attention. And what the people who built the vision transformer realized is that this idea of long range attention, so understanding how distant structures combine together to convey interpretation of an image, is also important for image understanding. So in this sense, they throw away all the inductive biases of a convolutional neural network. So convolutional operations are matrix multiplications of very small local filters. So the filters only have a very local understanding of the image. So they throw this away. So you have a very computationally heavy process where you have long range attention. So you know how different tokens in the image correspond. So it becomes computationally more heavy, potentially more prone to overfitting, but more powerful. So basically they just work by taking images, splitting them into patches, flattening the patches, and then pushing those patches through a conventional transformer with exactly the same architecture as a language model. And this can then be easily translated to other domains that are non-Euclidean simply by defining a patching system for those images. So what Simon did was he took the cortical imaging data, projected it to a sphere, which we're already doing for image registration, patches it using a triangular tessellation from a regular icosphere, and then does exactly the same thing, flattens them as a positional embedding so that you impose a little bit more of an inductive bias, so you do know which patches are neighbors of each other's, and then just pass it through these multi-head self-attention layers. Where the self-attention operation itself is just learning these query, key, and value matrices. So these are the weights that you're learning from your model, the parameters of your model. And then you're querying, your self-attention operation is just looking at the similarity of these queries and keys. So basically, how important is attending to a specific token in your image to another for understanding the structure of this image? They're then multiplied with the values such that as you go through each layer of the self-attention, the value for each token becomes a weighted contribution from all the other tokens, meaning that those tokens that are more important for understanding the structure of this one become embedded into its content. And this is directional, so you learn a query and a key such that, and that's important in language learning because For example, in this sentence here, Evan's dog Riley is so hyper she never stops moving. The understanding that the subject of the sentence is the dog is more important than really knowing what the name of the dog is. And so it's important for language learning, but I think it's also really useful for image understanding for medical images particularly, because it should probably make the model more robust to noise and inter-subject variability. So knowing that for example, in functional activations, knowing that some structure is firing up, certain combination of structures always should fire up together, is more important than, for example, if one structure fires up on its own and it's just there because of noise. So I have a sense that it's probably quite useful for dynamic modeling of function. The other advantage of transformers is that they're inherently interpretable. So you get out of your self-attention model, looking at the attention of your model, looking at the intention of the class token, so the token that goes into your prediction or your classification model relative to all the other tokens, gives you some understanding of what features of the image were important for making a prediction. So this is classification of how old a baby was when they were scanned, and you get a separate attention map for each individual. And this is a baby born at 32 weeks, or born early and scanned at 32 weeks, and another scanned at 42 weeks. And you see that the model is attending to different structures, particularly focusing on areas of the brain that are developing rapidly during this late period of gestation, so cortical myelination and increasing sulcal depth. The problem with transformers is the complexity of the self-attention operation scales with the number of tokens you've got squared. So you either have to use quite low resolution patches, which gives you a less interpretable self-attention map, and less ability to pick up on really subtle features of the data, or you use engineering techniques such as swim transformers, which learn a multi-resolution solution, where at the highest resolution you have very fine resolution tokens or very small triangles, where you're only learning attention within windows, and then you progressively downsample so that your windows become bigger, but your token resolutions also get smaller, so that at the end, you're attending to the whole image at low resolution, but in combination, what you get is much more sensitive prediction. So relative to surface convolutional neural networks, you get networks that are very sensitive to cortical phenotypes and transformation invariant. So here we're trying to predict how old a baby was when they were born from their term age scan. And the error here is much lower than a week. And when you consider that the error on the ultrasound dating is probably within about half a week, this performance is really good relative to what we would be able to do with classical image processing. You can also do segmentation. But the main motivations for building these models originally was to build models of brain functional dynamics. And so in order to do this, we first had to get the models to train stably and generalize well. And the problem with transformers is because they've removed all inductive biases and you have so many parameters to learn, they can overfit. But the way in which you get them to work is to do self-supervised pre-training. So you train a model to learn how to reconstruct an image when you've removed most of the patches. So you remove most of the patches, pass to an encoder only those patches that remain. So you learn a smaller encoder that can be deeper. It then has to really learn how to model the structure of the data in space and time for function. And then has a decoder which learns how to reconstruct these in full. So you can see from fMRI that when you're doing this, you're patching, you're removing 90% of the image. So you're retraining tubes, essentially what we call tubes, so small patches in space and time. So the space and time is consistent, but most of the cortical surface is removed. And even doing that, the model can learn to really reliably reconstruct the cortical functional dynamics. And that's because it really then has to learn through its attention model that if it's observing some pattern of activity here, And here, for example, that it is corresponding to this particular functional network. So it has to learn something about the spatially distributed functional networks within the brain. And then doing this, then you're able to start doing more sophisticated things. So on the basis of that video surface masked autoencoder, we were able to encode the functional dynamics of human fMRI during a movie watching task, align that data to video masked autoencoder encodings of the movie scenes and a audio transformer encoder, align these with clip contrastive learning so that we know that when we see a specific pattern of fMRI, it aligns to a specific structure in the image and the audio. And from that, then we're able to build generalized models of brain decoding. So where people would previously have built models within individual and across within individuals, so you would have had lots and lots and lots of densely sampled stimuli from hours of movie watching, and in order to be able to predict what scenes and in order to predict new scenes from that haven't been seen during training, but within subject. In this case, we're able to predict what scenes we're observing in entirely new subjects with really high accuracy. So here, the clip retrieval is saying that, can you predict what scene you're looking at from a list of 100 possible scenes? And the SIT does a much better job than a model that basically just, this one just takes out all the fMRI, removes all understanding of the spatial temporal dependencies or the spatial autocorrelation of the signal, and just changes it to a vector and tries to predict from that image vector. But not only can it generalize to new subjects, it can generalize to predicting scenes that it hasn't been trained from individuals that it hasn't been trained from. So this is the clip retrieval task. So this is the reference sample, for example, that it's trying to predict, and it has a whole list of different samples. And the thing about this is it's the HCP70 movie watching data set. So each individual movie only appears for a very short period of time, maybe 3 minutes. So it's very diverse, different types of movies, ranging from blockbuster movies to art house movies, and very short clips across, but across more individuals. So there's 170 individuals. Usually decoding data sets would have just maybe 8 to under 10 individuals, but the amount of stimuli would be hours and hours of movie watching, for example. So even with all of this diversity, it's able to reliably predict the right clip. And when it does, and the top clips are always have the same semantic content. And this is for new scenes from new subjects. So the data was, these models weren't trained at all on these data sets. The other thing that's useful putting this all together is that you can then look at the patterns of self-attention to see how the model is learning to encode this, and the patterns of the different heads of the transformer are encoding different functional networks from the brain. So the first to the fourth attention head are looking at sensory motor and auditory networks. The fifth and sixth are looking at visual networks. And while this might seem not unusual for a movie watching task, I think this is for those tasks where the mechanisms are less apparent or less clear, it gives you some way of understanding what mechanisms are going on in the brain underneath. We also did a little bit of generating the scenes purely from the fMRI data, and you can see here, this is from Inception. There's 2 people, Leonardo DiCaprio and I forget her name, walking down the street, and from the brain imaging data, you can generate 2 people walking down the street. But I think this is a bit gimmicky, but probably needs some work. And finally, I'm just going to say that moving from beyond just detection, I think that the direction of travel needs to be in embedding biophysical models within our learning frameworks, with a view to improving, basically learning a better biophysical model of the data, but learning a better biophysical model of the process from the data. So in this sense, the first thing that we've started to do is build biomechanical models of brain atrophy and Alzheimer's disease. So here, for biomechanical models, we have a hyperelastic strain function. We know from finite element models that we can do a reasonable job of modeling the biomechanical processes of cortical atrophy, and we can do a slightly less good job of modeling the biomechanical processes of cortical folding, but our understanding or our physics models of these are incomplete. Similarly, our physics models of brain functional dynamics, for example, dynamic mass models or dynamic field models, are incomplete. With learning models, there is a space within here to learn a better model of the data, a better model of the process from the data, because if we have longitudinal data or dynamic fMRI data, then we have data with which to constrain the learning of the physics model. So in this particular case, we've just got a relatively simple model where we've got a model of an isotropic model of cortical atrophy, and we're training the network to learn this model. So this is a prototype example, I guess you could say. So into the model just goes demographic data, the individual's age, diagnosis, sex, and an interval between two scans and their baseline scan. From this metadata, you use a regression network to just predict the atrophy of different brain regions, and then you project that into a segmentation image so that you've got a value, an isotropic value of atrophy for each location. You push this through a UNet and it learns a deformation field where the loss function here is purely a biomechanical loss. So the idea here is that the FK is the elastic deformation, so the deviation away from the prescribed model. So you minimize this separately, then put the networks together, and you're trying to train a model that learns to deform this baseline scan to look as close as possible as a ground truth follow-up scan, while staying as true as possible to this biomechanical model. And then when you go in, once you've done this, then you're no longer constrained by the exact time points you've got. You can intervene on the different properties and simulate different patterns of cortical atrophy, can intervene on different disease states, different time intervals. This is a very coarse model, but nonetheless, you can see that it does a good job of predicting the core patterns of cortical atrophy of the ventricles and the hippocampus sufficiently well that you can see that they distinguish clearly between the controls and the disease states. So that's it, basically. My pitch for improving the precision of cortical neuroimaging for biomarkers and precision medicine would be the following steps. So I strongly encourage, as always, surface-based analysis, because the cortex itself is a curved manifold, and understanding its properties is better done by doing it on the surface, as has been shown by all of the studies from FreeSurfer and HCP, etc. But in that space, we should be going, because of all this heterogeneity, there is a reason to go to learning-based solutions that are, because they're transformation invariant, they can be robust to the residual confounds that are really difficult to deal with through image registration, and I really don't have a good idea how to fix them because of the extent of which they are variable, and the fact that we don't fully know what the mechanisms are driving those. But beyond that, you need to go beyond just using deep learning to classify. You need to use generative or inherently interpretable AI models. Ideally, in the long term, the trajectory that our lab is going on is to build biophysically constrained models of a range of neurological processes in order to better understand them and potentially use them for prognosis and prediction of outcomes in disease. So as always, most of our tools are available through our GitHub. If not now, then will be soon. There's a new version of MSM if anybody uses it. I had a software engineer who redeveloped MSM and got it running in 2 minutes instead of the 90 that it used to take. So if you do use it, I suggest using that one. There's various tutorials on surface analysis and machine learning that we're putting up on YouTube. And I just wanted to thank my team and my funders. Thanks.